Hi, I'm Ben Cobb.
And I'm Conor Moran.
This year we looked at the effect of glucocorticoids, cortisol, and the chemotherapy aid dexamethasone on chemotherapy counterproductivity. Cortisol is a hormone secreted in the body naturally. It averages about 72 nanograms per milliliter, but a stressed out person is 216 nanograms per milliliter. Cortisol is secreted when someone undergoes chronic stress in the body to prepare them for action. Dexamethasone is a chemotherapy aid administered in 90% of all chemotherapy regimens. Dexamethasone is a aid to negate certain side effects such as nausea, and it has the same function as, or it has the same structure as cortisol. So cortisol and dexamethasone work hand in hand. We looked at chemotherapy. We found out that cortisol increases chemotherapy resistance, and we wanted to find out why this was. Now, we looked at cancer stem cells and metallothionein, and those hypotheses didn't work, so then we looked at multi-drug transporter resistance.
To test multi-drug transporter resistance, we used three methods: fluorescence microscopy, qRT-PCR, and quick cell proliferation assays. The fluorescence microscopy data showed a global effect of efflux of the hoax 33342 dye, which led us to believe that it was multi-drug transporters that were causing this resistance. We then looked into four known multi-drug transporter genes: ABCC1, ABCB1, ABCG2, and ABCC11. We saw actually up to a 580-fold increase in multi-drug transporter gene expression when MCF7 breast cancer cells were dosed with dexamethasone. We tested two cancer cell lines, MCF7s and neuroblastomas, to look at tissue specificity. We saw a large degree of tissue specificity. Ben is now going to go into the implications of our study.
Physicians must immediately stop and reevaluate the widespread use of chemotherapy aid dexamethasone because it's actually very bad and very counterproductive to chemotherapy. Also, cortisol and increased levels of cortisol is actually very counterproductive to chemotherapy. This answered a think tank question proposed by the NIH in 2005 of how does cortisol affect chemotherapy resistance at a molecular level. Thank you for watching our presentation.